Cancer remains the leading cause of death worldwide. But what is cancer?
Cancer is a disease that is characterized by rapid abnormal cell growth and division. Cells start to grow uncontrollably. These cells may form a mass called a tumor. The tumor may be populated by both cancer cells and a variety of ostensibly normal cells, such as cancer-associated fibroblasts.
Well, how do we treat cancer?
Cancer has a variety of treatment options, such as chemotherapy and surgery. However, we'll be focusing specifically on radiotherapy. Radiotherapy utilizes ionizing radiation in order to either kill cancer cells or damage their DNA, which slows down their growth. Radiotherapy induces the breakage of the double-stranded helix structure within cancer cells, resulting in cell cycle arrest, senescence, or apoptosis. Ever since the discovery of X-rays in 1895, the field of radiotherapy has had significant advances and is currently being used to treat more than 50% of all diagnosed cancer cases. Neoadjuvant radiotherapy is typically given prior to the main treatment to shrink the tumour. It is given alongside chemotherapy and plays a significant role in the management of locally advanced tumours, including rectal cancer, which we will be focusing on today.
So the overall aim of radiotherapy is to induce damage on the tumor cells?
Yes. As a result, early radiotherapy research prioritized killing the cancer cell itself. This is through strategies such as increasing the dosage and image-guided RT. These allow accurate targeting and enhanced RT delivery through more precise dose deposition in the tumor, while limiting its delivery to vital normal structure. However, this completely disregards complex biological interactions between the tumor and environment that it colonizes.
So this is where cancer-associated fibroblasts come into play?
Exactly. Cancer-associated fibroblasts, or CAFs, are one of the most abundant stromal components in the tumor microenvironment. In rectal cancer, they form part of desmoplastic stroma, which is the fibrous connective tissue around the tumor. CAFs have an active role in tumor progression and cancer pathogenesis by offering radioresistance through the production of inflammatory cytokines, regulatory factors, and other tumor-promoting growth factors. This makes radiotherapy treatment surrounding cancer-associated fibroblasts revolutionary by focusing on the role of the tumor microenvironment rather than just the cancer cell.
Okay, so how does this radiotherapy CAFs treatment work?
In response to paracrine signals, radiotherapy-activated CAFs induce DNA damage via triggered p53 activation, cell cycle arrest, and the secretion of paracrine mediators, including insulin-like growth factor 1. Collectively, these factors promote the survival of colorectal cancer cells.
So how was the effect of radiotherapy on cancer studied?
Scientists use orthotopic colorectal cancer models, which involve the use of mice to mimic human colorectal cancer development process. It enables the study of cancer growth, invasion of the tumor, and liver metastasis.
Okay, so what experiment are we focusing on today?
Well, we will be focusing on the functional role of paracrine insulin-like growth factor 1 receptor activation by irradiated cancer-associated fibroblasts. More simply put, this in vivo experiment investigated whether the response of colorectal tumors to radiotherapy is improved by the neutralization of insulin-like growth factor 1 receptor.
Okay, so how can these receptors be neutralized?
In order to neutralize the growth factor receptor, the human monoclonal antibody R1507 was used, which is currently being studied due to its possibility to kill cancer cells. R1507 is able to do so by first binding with high affinity to the extracellular alpha subunit domain of the insulin-like growth factor 1 receptor. This prevents the insulin-like growth factor 1 ligand from binding to the receptor, stimulating both internalization and degradation of the receptor. The result is the neutralization of the insulin-like growth factor 1 receptor.
Right. And how was this experiment conducted?
In this particular experiment, mice were first injected randomly with either HT29 or HCT8 cells, which are cancerous cell lines. 2 weeks later, a CT scan was conducted on the mice in order to localize the tumor. Mice that had an equal intensity of cecal bioluminescence received 10 GY of irradiation. After another 24 hours, mice were then randomly selected to be injected with either a placebo of intraperitoneal saline or a H1507 monoclonal antibody treatment. Next, mice are injected with luciferin 10 minutes before bioluminescent imaging is carried out. In this experiment, bioluminescent imaging was carried out bi-weekly in order to assess and analyze tumor development.
But why is bioluminescent imaging used?
Bioluminescent imaging is the introduction of luciferase, an oxidative enzyme that produces bioluminescence into cancer cells within the mice. The cancerous cells will glow, with the brightness depending on the abundance of cells that are growing and dividing. Therefore, it can be determined that mice with a greater bioluminescent brightness will have a larger amount of cancerous cells within the body.
This investigation concluded that IGF-1R neutralization following radiotherapy through the use of R1507 monoclonal antibodies reduced the number of mice with organ metastasis, thus improving the outcome of colorectal cancer in vivo. However, this experiment does possess some limitations. For example, the use of a greater sample size of mice would increase the reliability of the results obtained. Furthermore, the results may differ when tested on humans. Overall, the technique of this experiment was very effective in determining the effect of IGF-1R neutralization.
Cool. So, what did the experiment actually find?
In the experiment, there were three key findings. Firstly, the activation of IGF-1R signaling in response to paracrine factors such as IGF-1 leads to downstream signaling cascades, including the activation of AKT, a kinase involved in cell proliferation. Figure 4D, the Western blot analysis, illustrates the presence of phosphorylated IGF-1R bands. This suggests that the increased phosphorylation of IGF-1R could be in response to paracrine factors that activate IGF-1R signaling.
Secondly, it was found that the neutralization of IGF-1R activity by monoclonal antibodies such as R1507 blocked the increase in cell number, lactate release, and glucose uptake by cells. Figure 5C denotes that R1507 was able to inhibit the concentrations of extracellular lactate. Figure 5D shows how the inhibitors also successfully counteracted the increase of glucose uptake.
And finally, R1507 following radiation significantly reduced the number of mice with metastasis, as it not only stimulates cell death in colorectal cancer cells in laboratory settings, but also leads to improved outcomes in live animal models. So, the best way to reduce mice with liver metastasis isn't radiation-only treatment, but the use of R1507 monoclonal antibodies following radiation. The figure shown displays how R1507 significantly reduced the mice with liver metastasis compared to the radiation-only treatment.
Okay, so the results from the experiment indicate that paracrine activation of the insulin-like growth factor receptor by radiotherapy-activated CAFs promote tumor progression. So these findings can have substantial implications in the real world. The pro-oncogenic functions of CAFs make them an attractive therapeutic target for cancer treatment. This could include using high-precision radiotherapy to provide a large radiation dose to these CAFs in the tumor stroma, while sparing surrounding healthy tissue with much lower doses to reduce toxicity. CAFs have also been determined a promising clinical biomarker for diagnosis and prognosis, with potential roles in predicting the therapeutic efficacy and responses of particular cancers. But can this research be applicable to other cancer types?
Yes. Whilst this experiment focused on the role of CAFs for colorectal cancers, there is promising research regarding the applicability to many other cancer types. This is due to tumor stroma and CAFs being a consistent feature amongst nearly all solid neoplasms. This is shown in the table, which highlights the role of CAFs in cancer pathogenesis for cancers such as prostate, cervical, and breast cancers.
Ultimately, these findings have an important contribution to broader society by assisting in diagnosis and treatment options for several cancer types with potential to increase cancer prognosis and survival rates.
However, research targeting CAFs has faced several obstacles, including the lack of specific CAF markers, posing a challenge to precisely target the fibroblasts without interfering to some extent with the neighbouring cells. As well as this, the diversity of CAF markers and tumour environments pose an obstacle for standardised treatment and highlight the need for more large-scale studies for different cancer types. Thus, before clinical application, greater research is needed to determine the precise function of the identified CAF biomarkers, as well as their differing roles in cancer pathogenesis.
So radiotherapy-activated CAFs could potentially be the future of cancer treatment.